let's continue with the seminars. Now it's time for the short talks. And the first talk will be by Rebeca Bocanegra. Rebeca is researcher at IMDEA-Nanocience at the Molecular Motor Manipulation Lab with Borja Ibarra. Rebeca obtained her PhD in 2011 in the molecular biology field. Since 2016, she has been working at IMDEA-Nanocience studying membrane behavior. Rebeca, if you are
Yes, I'm here.
available. Okay, so you can start now.
Yeah, thank you for the nice introduction. So I will share my screen now. Can you see it?
Yes. Yes.
Okay, I will show you today this work that we have recently published about the dynamic constriction and fission of endoplasmic reticulum membranes by reticulon. Well, as you all know, the endoplasmic reticulum is a continuous membrane system inside the cell, which is highly dynamic and is constantly changing in the cell cycle, and has some parts with highly branched tubules and parts with flat sheets, and it has interactions with cytoskeleton, plasma membrane, and other organelles. And the maintenance of this system is related in one hand to atlastin proteins, which are fusogenic, and on the other hand has been related to reticulon. Reticulons is a family of proteins which share a common structural feature, which is a hydrophobic part, which is able to form a wedge-shaped hairpin that spans on a monolayer or the whole bilayer of the endoplasmic reticulum and can interact with the cytoskeleton. So among the evidences that say that reticulon is involved in the endoplasmic reticulum maintenance are those from the group of Arturo Dagas, who is one of our collaborators, that have seen in animal model in Drosophila, which only has one atlastin gene and one reticulum gene, that if you avoid the expression of reticulum, you obtain this endoplasmic reticulum, which is without any branches, and with an overexpression of reticulum. The endoplasmic reticulum, I don't know if you can see here, this red dots, it's highly disconnected. They have expressed in COS7 cell this reticulum 1 gene, and in this case, they have seen the disconnection of the endoplasmic reticulum branches, and they have been able to observe the fission of some of the branches of the ER, and this fission is always occur in the zones of the ER that has been remodeled in active remodeling, of course, and this retraction of the branches to the ER is slower when with this overexpression. And the goal of our investigation in this part is to see with our technique, which is optical tweezers, we wanted to test the fission activity of reticulon and characterize the mechanics of this activity. For this, as I have said, we use the optical tweezers technique. We have two dedicated mini tweezers machines which consists in two counterpropagating laser beams focused in the same point to generate a single trap. This is all inside a microfluidics chamber with a pipette, so we can attach our biological system of interest between one bead, which is held by suction in top of this micropipette, and another bead which is in the optical trap. This optical trap, we can move it up and down, so we can exert force in our biological system, and we can measure the forces too. So to study the membranes, what we do is to generate, I will pass it fast, generate some vesicles on top of these plastic beads, and generate between the two beads a lipidic nanotube. How we do this is, for example, with a pure lipidic sample, what we do is to put together the two beads and pull the two beads apart. This generates a nanotube, and we can follow the formation of the tube by the characteristic Forks extension curve that is recorded by the machine. In a pure lipidic sample, once you obtain this nanotube, the force that we measure is constant and is reversible and is related with the radius of this tube. In the case of the reticulon lipid sample, we have the reticulon. It's a proteolipid sample, sorry. So we do exactly the same, but the force extension curve that we obtain is quite different from the pure lipidic sample. We see this increasing force as we stretch the tube, and as we put closer together the two beads again, we see a huge hysteresis. So we have done this kind of experiments with the pure lipid samples and reticular lipid samples. We at different pooling rates, and what we observe is that we can do a lot of different consecutive force extension cycles with the lipidic samples, but with the reticulum samples, at some points these tubes break. Always when the tube is in movement. Of course, as we are stretching the reticulum tubes, we reach higher forces than in the lipidic samples because of this kind of behavior. But even if we pull with really high forces the lipidic tubes, we never see fission in the lipidic tube. Sorry. So here I show you the increasing force that we obtain in the reticulum tubes in the moment of fission, which is the black dots, and the maximum forces measured in the lipid tubes that I repeat that never get broken. So, and the forces are quite similar in the higher speed range. So the high forces that we obtain are not the only reason that gets the tube broken. So we can relate this increasing force in the case of the reticulum with reduction in the radius of the tube, because as I have said, the forces are related with the radius. In fluorescence experiments that were performed in Bilbao by our other collaborators, Vadim Frolov lab at Ikerbasque, they have seen that in static tubes, the tubes that they obtain in the presence of reticulum have these constricted and bulged parts. So of course, reticulum is producing a constriction of the tube, as we have observed in the force traces. Interestingly, in these static tubes, the reticulum is stabilizing, is counterintuitive, but the reticulum is stabilizing these constricted parts. And this we know because if you just, I don't know how to say that, if you just stop pulling the tube, or if you put together the two bits in our experiments, for example, you see this huge hysteresis and this huge decrease in the force. So reticulum somehow is stabilizing these tubes. So we have these two different behaviors here. In one hand, reticulum is constricting tubes, but is stabilizing the tube. This is related with the observations that in vivo, is mostly you can find reticulum mostly in the nanotubes of the ER and in the junction of the branches. The other observation that we have made in fluorescence experiments is that the proximal part to the reservoir, to the membrane reservoir, the tubes are even thinner, more constricted than in the distal part that is down here. From images, from fluorescence images of the tubes and the forces that we obtain in the same samples and at the same forces and pull speeds, we have calculated the bending modulus of the tube itself, and this bending modulus of the tube is lower than the bending modulus of the membrane, the bulk membrane. This is in our model. This is produced by the sorting of reticulum from the membrane reservoir to the tube, which is producing a viscous drag. If we represent the radius of the tube obtained from the forces in our optical tweezers experiments in front of the velocity, with the bending modulus, we use the bending modulus of the bulk membrane, we obtain radius that are, well, these two points in black are at higher velocities. This radius is below the minimum limit for the fission of the tube, but at low forces that are more comparable with the physiological ones that the cytoskeleton can exert. This radius is higher than the limit, but if we use this new bending modulus that we obtain from the thinner part, we can see that the nanotube radius is below this hemifission limit. So the conclusion is that the radius, the reticulum, is constricting dynamically the nanotube. So in conclusion, what we have observed is that reticulum 1 is using two synergistic modes of membrane curvature, static and dynamic. And the sensitivity to the membrane movement suggests that this dynamic regulation of the topology of the endoplasmic reticulum that links fusion and fission with membrane motility. So, and in the cell, this motility is driven by the cytoskeleton. Then that's why reticulum is that's the mechanism that reticulum is using to promote fission of the ER. So finally, I just want to thank all people in our lab at IMDEA-Nanocience and our collaborators in Bilbao and in Italy. And thank you all for your kind attention. I think I have talked too much, too fast maybe, but I hope you have more or less follow me.
Yes, thank you, Rebecca, for the nice talk. Virtual applause. So